you want to like in this world, it's better to have a potassium inside the cell. Okay. Bubble is opposite direction, but even in water, it may form, if it forms the vesicle, it should be same way of the double the layer. Okay, modern life, we developed it further and changed the membrane a little stronger in terms of hydrophobicity and hydrophilicity. Okay, good. Whether you memorize it or you calculate it, anyway, it's good to know about the 4 nanometer of the thickness is our membrane of the cell. Somebody has a little different opinion, but it is 4 nanometer. Okay, kernel size of the cell is approximately 10 micrometer. This size, 150 micrometer size of biggest one is egg cell. And length of a sperm cell, about 50 micrometer. Okay? But other than that, regular size of the cells, most of them in our body, about the 10 micrometer. Okay, it's easy enough to memorize. It's about 10% of our cell. Bacteria is about 1 micrometer size. Yeah, they say mitochondria came from bacteria, right? Inside our cell, bacteria is like a 10% in terms of thickness, not length. Bacteria can be a little long, right? So that is about bacteria size. Yeah, easy enough to memorize. That's also 0.1 micrometer. So that's an approximate number. But it's very convenient to memorize. Of course, there are variations. Some bacteria recently found it's bigger than some virus is bigger than bacteria, they say. But still, the average of majority of those viruses is much smaller than bacteria. And our cell is 10 times higher, bigger than bacteria. Okay, mitochondria, chloroplast, and nucleus is known. They have a double layer of bilayer, right? Double of a double. So we'll see those structures. mRNA is synthesized inside the nucleus by copying the information from DNA to make mRNA. Protein synthesis happens on ribosome, in ribosome, or on ribosome, and ribosome is mainly localized on ER. Mainly we can say that's the ER. Okay, partially it is modified on ER2, but it's a further modified main modification happens in Golgi. 생각 좀 더 하고 실전도 좀. 지금까지 정답이 너무 많았는데 내가 저기서 열심히 보고 있어야 문제까지 맞추면 Okay. So we are going to think about this further. In the video I gave you, there was protein synthesis and then moving. There was some meaning there. It was not randomly shown. Okay. That part we are going to study further today. Okay. Start. 뭐 장사가 너무 안 되는데 이거 줄게요. 잠깐만. 1등 했으니까 한솔. Okay. Okay, today we are going to study further about the structure of cells. Our cell, most likely our cell, our life studied in water environment. Okay. In water it was a form. So as we studied the last week, the life forms may emerge from water, and maybe the first step was isolating out our body from environment. Okay. So to do that, we utilize, or the life utilize, lipid bilayer. So lipid molecule with a charged polar group, and this is the first step. So this is me, this is environment. This is the inside space, this is outer space. Right? Inner space and outer space. And then to keep that as the identity, somehow we need to tell information. This is my information. Maybe in the beginning there may be simpler form of information, like some kind of a polymer which cannot penetrate this membrane. And anyway, it was developed further and further. Maybe RNA as intermediate. Now we use DNA as our genetic information. Still some life forms, like some RNA virus, if you consider like those life form, they still use the RNA as genetic information. But mainly we have either DNA or RNA. In the beginning, those informations are just captured inside of this membrane, but later, this is so important. We want to protect it by extra membrane inside. So that is what we call nucleus. So nucleus is formed, so there is a life forms without the nucleus and real nucleus. Without the nucleus, we call prokaryotic. Karyotic means nucleus. Now karyotic means nucleus of DNA, and pro means the before nucleus. Eukaryotic, EU means real nucleus. Okay? Prokaryotic, eukaryotic. So after that, to get better energy of the generation, they accepted some power plant, mitochondria. And mitochondria, we believe that it was a separate bacteria which has developed the smooth oxidation reaction. And then it has so powerful and so efficient to generate more energy. And this kind of very dangerous power plant, the coal-based power plant, like a power plant, So it's difficult to keep that inside the cell. And developing that kind of thing is a lot costs a lot of energy, and also it's potentially dangerous. So maybe it's better to accommodate, allow them to immigrate to inside our body, and then live together, so-called symbiosis. So that's why the mitochondria has its own gene. We have our own gene. They are different, and it seems like they are from the different origin. That's why we assume mitochondria originated from different bacteria. Also, we developed a small vesicle area inside the cell. So some of them, like a lysosome, they can burn out or remove those waste material. And to make a protein, rather than floating around the separate ribosome, so forming this kind of another the area of organelle, so-called the ER, endoplasmic reticulum, and then ribosome can be attached to the surface here, and they can make the protein of them. And then further modification step, we make Golgi body like this. This is kind of current the so-called our level of advanced cell types, eukaryotic cells, with a real. So for the membrane, in the beginning, last week, as we have seen, maybe the first form of a membrane structure is very simple fatty acid like this. But this one, as you can see here, this is a single chain, and this is carboxylate, which is relatively weak acid. So those forming of this double layer, the power may not be so strong. So this is the main structure from lipid molecule we can get from the animal fat, and by adding sodium hydroxide we can make a salt, right? But that salt, so-called natural salt, is less effective than synthetic detergent. So this is the structure of the natural salt. By getting triglycerol ester of this and then clean that by sodium hydroxide, we can get this. But most of those synthetic detergents that we use are stronger charges than this. So comparing to carboxylate, sulfonate is a stronger acid, and means it has a stronger negative charge there. So this is kind of the structure we use for detergent, and all of them is a negatively charged. Sometimes it's better to form positively charged one. This is so-called reverse the salt. Then it can disrupt the bacterial membrane better. So for the disinfection, usually we use something like this. Okay. Or sometimes not only the negative or positive. So sometimes we don't want any charge. Neutral but water soluble molecule, like the 2 carbon, 1 oxygen, 2 carbon, 1 oxygen. This is so-called ethylene glycol molecule. Then this is also kind of neutral. So lipoforms, by the way. So to form this membrane structure, we have to develop that into two tail, and then head group is a phospholipid group. This is the current form of the phospholipid we are currently using. And at the end over here, it can be different the structure on top. But anyway, up to here, glycerol is a two tail make a more hydrophobic, and better embedded inside here, and stronger charges. Phosphate can be two negative charges, so even stronger than sulfonate. So that is current form of a membrane structures. So let's first, not only single membrane, I would say first of all, this is a boundary of life. It's a very compact, there is no hole, it's a compact membrane structure, inner space, outer space. Right? But not only that single membrane structure, actually they form different vesicles inside. For example, let's say endosome, zone means body, endosome means inside, body inside the cell, endosome through endocytosis. So this membrane structure can be embedded inside, engulfed it, and then it forms a separate structure like this. Now this is called as an endosome, and this process is called endocytosis. Now if you look at those structure, this is inner space, correct? Ribosome. How about inside of endosome? Is it outside or inside? If you look at how it was formed here, that space inside of this lysosome, is that inner space or outer space? 자신이 없는 거야, 샤이한 거야, 아니면 겸손한 거야, 겸손한 척 하는 거야. See, that's outer space, isn't it? Even though this vesicle is inside our cell, but funny thing is, this membrane is also lipid bilayer, same composition like this. But inside of lysosome, or inside of endosome, is outer space. We keep the sum of those outer space in our inner space, right? And there's another process called exocytosis. It's the opposite process. Some of the endosome goes back, reverse reaction, and then it is secreting some material to the outside. And also, this is the endosome is that there is another, the vesicles, which is secreted to the outer space. That's what I call exosome. Endosome is the inner body, exosome is the outer body. Makes sense, right? Then exosome, how can you make? Through what process? Through exocytosis? You are secreting some membrane. What do you call that process? Exocytosis, okay. I said this exocytosis is a reversible reaction to this. If you had this kind of a form, if it goes this way, does it form any exosome? No, right? Exocytosis is already existing endosome is go into the membrane and then fuse, and then only inner material secret outside, but there is no extra vesicle form, okay? And that cannot be exocytosis. You need to have a different process. So by the way, if there is this kind of endocytosis is formed, then this is coming in and then form this kind of vesicle, or if this vesicle is becoming bigger and bigger, and then make a folded the bag like this, folded the jumani like this, then this can be either Golgi or ER. That means ER or Golgi is also kind of a very big vesicles inside. Right? That means inside of ER, inside of a Golgi, that's inner space or outer space? That's outer space. Right? That's outer space. That is very important to know to determine when the protein is synthesized, where it goes. It's inner space or the outer space. Okay? Now, let's see. This is the process. This is the membrane. This is inner space. And it should have opposite process of endocytosis. Endocytosis was the inward engulfment, right? So exocytosis, but in this case, it should push it out to the outside, and then it can form these kind of things. So this process is not exocytosis. This process is extra structures coming to the outside. This process is called ectocytosis. So this is different from exocytosis. You understand, right? Reversible reaction of exocytosis. What is it? Is it exo or ecto? Reversible reaction means exactly the same process, just going different way, right? Reversible reaction of endocytosis is exocytosis. Ecto is the result is different, right? That's a different reaction. Computing? If you look at here, this is endocytosis process. Reversible reaction is this goes back, right? That process is exocytosis. So exocytosis is a totally different mechanism. That means different protein machinery should be involved here. So this through this exocytosis now it can form exosome. Now, next question. Inside of exosome is inner space or outer space? That should be inner space. It is secreted to the outer world. It's kind of sending satellite or the small fragment of our cell to the outer space. But that inside material is the same as cell material. That's inner space. Right? Endocytosis, by the way, comes from the environment. Even though they exist inside our cell, but that inside of that vesicle is outer space. Same as ER, same as the body. Okay? So that's important to know. Now, So I give you this question, and you get a couple of answers here. And yeah, correct answer. Inside of the endosome is outer space, and inside of the exosome is inner space, right? So, and also synthesis of protein. mRNA is synthesized in nucleus, and then is transported to cytoplasm, and that information is read by ribosome. Ribosome is on the surface of ER. On the surface means ribosome exists in inner space or outer space. Do you agree? Ribosome is in inner space or outer space? That should be inner space, right? That's on the outer surface of ER, not inner surface, right? So that should be inside the zone. That makes sense because we should make a protein in our world, not outside world. Okay, ribosome is the inside the zone, so that's the inner space, and then you make protein, and depending on those process and also those locations, now those can go to different regions. So if you summarize those protein synthesis, first step is information to make a protein. We call it gene. One gene is the information to make the one protein. Of course, there's a classical definition of gene though. Anyway, that's one gene. The gene altogether, we have 25,000 genes in our DNA. Altogether, the collection of all genes, what we call. That's a genome. Or in original pronunciation in German, genome. Okay? The genome means it has all collection of all genes. That's the genome. Okay? Now, some of one gene is transcribed into RNA, and the machinery to make this protein is ribosome. By the way, ribosome is what is the material to make a ribosome? It's RNA, not protein. Very interesting. Most of those calories in our body, enzymes are protein-based enzymes. But in a sense, most fundamental and most important calories, making protein, it should be fundamental, most important basic function, right? That is made by RNA. So that's why there's some hypothesis that maybe ancient times, before we have DNA, protein, and separate functions. Separate function means the DNA is an information system, like a magnetic tape, or just a disk. And protein is the real machinery system, like the functional program. In the middle, there may be some form of RNA can do genome and also can do protein functions. That hypothesis is RNA world hypothesis. It doesn't exist anymore, but there's some traits. For example, some leftover other traits like ribosome is RNA, but it's a functional name. And it should make a protein. After that, it should decide where to go. We need the proteins everywhere inside the cell. Inner, outer, every side, we need those proteins. And then sometimes protein is based on 20 amino acids. It has a diversity, but sometimes we want a little more diversity. Then we modify that further. That's what we call post-translation modification. Translation is the process of making protein. Before that, from DNA, making RNA, what we call transcription. Okay. Transcribe is the writing, translate is the translate. Okay. By reading that and then transfer that information to the product is called the translation. Transcription and translation. Okay. Then now we need to think about where to go. We need those proteins inside the zone, definitely. Some of those proteins we use them as secretion, like hormones. Or some of them is bound on the membrane. It's a different kind of the destiny is there, and further detailed explanation will hear. Please invite her. It's okay, invite her. Okay,
my name is Suhyun-sa, and today I'm gonna talk about the protein synthesis, and especially protein sorting. Like the professor said, the protein is made like this process. At first, the genetic information in DNA is transcribed into mRNA, and after the mRNA modification inside the nucleus, it goes out of the nucleus with the nuclear pore. And it goes to the ER, and some of the post-translational modification is occurred at ER, and through the transport vesicle, it goes to Golgi, and it finally gets out of the cell, and it is just about the secretory proteins. But there are also non-secretory proteins too, such as proteins used in organelles. So I had this question about how can these proteins move to proper location. Like in the real world, think about the mail delivery. The mailman see the post and the address is on it, and he can bring this post to the each location according to this address. So in the cell system, does it work same? And the answer is yes. The signal sequence is the address inside the cell. So as you see in this picture, there are signal sequence of each organelle, and according to this signal sequence, the protein is moved into each organelle. So in this presentation, I'm gonna tell you how this works. And as I said, we can categorize the proteins like secretory proteins and non-secretory proteins. And as we learned at our high school, the secretory proteins is made with ER-bound ribosome, and non-secretory proteins is made by free ribosome. But actually, these two are the same as structural or functional way. So what makes this different? The signal sequence and the SRP, which is signal recognition particle, make these two ribosomes function different. So I'm gonna tell you about the more process. When mRNA, this is about the secretory proteins, and when mRNA comes out of the nucleus, the ribosomes assembles to mRNA, and the translation is started. And since the signal sequence is at the end terminus of the whole sequence, it is translated first. This red part is the signal sequence, and the signal recognition particle recognizes it, and then it brings this whole complex into the ER surface. So at the ER surface, there is a SRP receptor and protein translocator. And after the docking is finished, the translation gets restarted, and the proteins go inside to the ER. So what just I explained is about the co-translational translocation. In the case of secretory proteins, the translation and translocation occurs together. And on the other hand, on non-secretory proteins, the translation is finished at the cytosol, and then it is moved into each organelle. So let's wrap up with this picture. For secretory proteins, when mRNA gets out of the nucleus, the free ribosome starts the translation. And if it has the ER targeting sequence, SRP brings this whole complex into the ER, and then, like we all know, these polypeptide chains go into Golgi and then it is secreted. And how about non-secretory proteins? After the translation is finished at the cytosol, according to their targeting sequence, they move to nucleus, mitochondria, chloroplast, or peroxisome. And if it does not have the targeting sequence, it remains at the cytosol. And this is the end of my presentation, and thank you for listening.
Questions? Yeah.
I have a question. Your presentation, the co-translation of case, if the ribosome failed to define with some rough ER, if the co-translation is if the translation is just stopped, or
You mean fail to
that ribosome, if an RNA has some signal sequence, if SRP bind it and ribosome and SRP just bind with ER, and then they do some translation. And if just we suppose that ribosome and ER fail the binding, and how it works, just stop translating.
Maybe something goes wrong. I will come back to that later. Any other question? Okay.
oh
But
So when the protein is made in the cell and it isn't going outside, it's not being secreted, how does it move around the cell? Does it still get put in a vesicle? Vesicle?
I mean after translation, how can it move to these organelles?
Mm-hmm.
For the nucleus, there is protein named important, and it moves this polypeptide chain into the nucleus. And in the case of mitochondria, the chaperone with a protein named chaperone use ATP to bind to the peptide and chaperone, and it moves to the mitochondria.
okay, getting more and more complicated. Let's stop here. Okay, good. Okay, let's look at a little simpler version of this. I tried to make, and let's try to answer those questions. Okay, if the protein is synthesized like this, so on this, maybe the ribosome can be free ribosome, or maybe on the surface of a ER. If the protein is just synthesized by reading this mRNA and just synthesized and secreted here, then this should be in the cytosol. Okay? That's in inner space. Now it can be either floating around free protein in the cell, or it may have some signaling of the peptide, this end or this end. And depending on that signaling peptide, SRP signaling recognition protein come in and bind here and carry that to other localizations possibly. Okay. So if you show the one example, that very early stage of the synthesis, SRP, maybe this is the first part of them, SRP bind here, it affects the synthesis process, that's one possibility. Or it's a fully synthesized and then SRP bind and carry. If they fail those original the intention, they may do something wrong, either stop in the middle or they move to the other place, different possibilities. So this is simple one scenario. Protein was synthesized and secreted to the cytosol, inner space. So that's one possibility. In those movies we have seen, those protein, this ribosome was here, the synthesized protein was secreted inside the ER. That was the movie you have seen. Then if it is secreted in ER, is that outer space or inner space? That's outer space. It's ready to be secreted, right? And then this vesicle, originally this vesicle is also part of this ER, maybe. It forms this kind of a process similar to what process? Ectocytosis, right? Similar to ectocytosis. But morphology-wise, topologically, this is the outer, this is the inner space. This may be exocytosis, endocytosis, right? Because it's inner space, it's outer space. Anyway, it forms this kind of vesicle, and it is moving to the surface of the cell. And there was some carrier, right? Carrying those vesicles and move around, following which structure? That on the bottom it was working on surface of what? What is the name of the cytoskeleton? Microtubule. Cytoskeleton, there are many kinds, but the biggest two classes is, one is microtubule, it's going outside. It's suspending those cell structures. Another big one is the actin. Actins are surrounding the cells. So actins, the force is suppressing to the center of the cells. Microtubule is suspending. Okay? So if you look at any cell pictures, if there are any structures putting outside, that's microtubule. If it's the surrounding cells inside the membrane, that's actins. Okay? So along with the microtubule structure, this motor protein carries what? One protein plus a whole vesicle. This vesicle was carried by this motor protein. That's why we have seen. Okay? So if there is a secreted into ER, there is a secreting protein. And eventually it goes to the cell surface, and through exocytosis, the content of this protein will be secreted to the outside. Makes sense, because this is anyway outer space. Outer space material goes to the outside. Right? In any cases, we didn't see so far mixing of outer and the inner space together. We didn't see that, right? Outer space was always outer space. Inner space was separate from outer space. Right? Another scenario is this protein is secreted into this area, but it is not fully separated. It is still bound here. The end of position here, the last the peptide is synthesized. It has binding motif and it's bound here. Then there is some anchor. Anchor is on the big ship. We have this kind of structure, right? This is the anchor. It's holding that protein on the membrane. And the majority of this protein is facing inner space or outer space. Majority of the synthesized protein is facing inner space or outer space. It's outer space, right? Only the anchor part is on the membrane. That means if this vesicle moves to the membrane, plasma membrane, and eventually the protein on the cell surface, is seen outside or inside? It should be outside. That is the result of this. Then this is inside the cell, this is outside the cell. This is the three scenarios. Then my question, how can you make the protein not facing outside? Sometimes we want to have that protein facing inside, right? How can you make it? Out of those three scenarios. Scenario one, the protein was synthesized inside the cell. Scenario two, secreted inside ER in outer space and secreted. Scenario three, synthesized into ER, but it was anchored on the membrane. But that protein should be facing outside eventually. Now, if you need any protein facing inside, how do you do it? Scenario 1 to 3, which one? It should be scenario 1, and then in scenario 1, maybe end up here, there is a sequence, signalling sequence, and then it can be carried by SIP. This SIP can deliver this to any different conditions. After it reaches its the final destination, most likely it will be ending on the membrane. Then majority of the protein is facing cytosol, and synthesis and also itself delivery to the body. Of course, in real cases, it's much more complicated. Case by case, it's very complicated, but this is a simplified 3 scenarios. Okay. Now, if I ask you the question in the midterm exam, how can you design those delivery of the protein to the membrane facing outside or facing inside? Or I give you this picture. Okay, if this is the situation, what can be the end result of those localization? Okay, so this is 3 scenarios of the protein, and also in terms of the topology, either inside, inner space or outer space. Okay, based on this, there's a thing. Membrane. Our plasma membrane not only just a segregating boundary, it's an active function on the membrane. One of them is potential. Potential is applied to the membrane. Usually outside is partially charged, then inside. Now if you look at those phospholipid structure of a membrane components, phospholipids, 2 tail, and glycerol, and phosphate. If it is a simple phosphate, that's a -2. Sorry, if it is a 1 proton and the attached to form, this is most of them, it's a -1. The other position is attached to this, -1. But in many cases, it can be modified by different structures. If the modification is neutral molecule, this is minus plus, it's altogether neutral. Anyway, these guys are all -1. If that position is modified with a positively charged one, ethylamine or choline kind of a structure, then some of those charges should be negative or positive neutral. So phospholipids, majority of them is a negatively charged. Some of them is a neutral. There is actually almost no positively charged phospholipid. If it is all negative, then how can you make this positive? Sorry. By ion concentration, that's the next step. But if it's without the ion, if you form inner and outer, if they may have a different composition of this phospholipid, then how can you get positive? Even
using DNA minus charge.
DNA minus charge. DNA is trapped inside the nucleus and it's far from this membrane,
Use RNA instead.
sending RNA to make a. But RNA is also negatively charged. How can you make a positive charge?
Outside.
So sending out the RNA to the outside, then it's also negatively charged. How can you make a positive charge? You wanted to make a positive charge RNA? Amino acids, sending out amino acids, you already said the ions, so that's the next step. Actually, there is no positive charges. Inside and outside, composition is slightly different. Actually, not slightly, it's very different. It's not symmetric, it's asymmetric. Inside is a more negatively charged phospholipids. Outside is maybe more neutral. So relatively, outside is positive than inside. Relatively, relative potential is outside is positive than inside. It does not mean that the surface area, either inside or outside, is really positive. Charge-wise, both sides are negative charge. That means if you add any negatively charged molecule to the surface here, and positively charged molecule here, which may bind to the surface better. Cross charge, it's a attraction, right? Negative charge will be the first on the surface. That's why most of those drugs we use positive charge, not negative charge, because the effect is not so good for negative charge. Neutral is a neutral. Neutral is fine, but either positive or the neutral one can penetrate. The negative one has much more difficulties. Okay. Now through this. Next step is negative charges only, it may not be happy situation, energetically unstable. That's why count cations in both sides and neutralize. If it's just water, then more negativity inside than outside, like that. Now, the cations outside is more sodium plus, and inside is more potassium plus. Depending on that concentration of sodium and potassium, can be charge balance, it can be reset. If it is more positive here, then it can be neutralized slightly more positive constantly. Now this is kind of a balance in both sides. And the result is still this is positive and this is negative relativity. And in many cases, especially in neurons, we need to send out the signal by changing this potential. To do that, we utilize this channel here and send those ions from one side to the other. And very small amount of change is not huge. Like just 1% of the potassium is moving from one side to the other. Still, it can change the membrane potential. Then there can be signal, and this is electric signal. It can go very fast. Theoretically, it can go maximum to the speed of light. You can transfer. So that's the speed. We see something, and then touch or hot, kind of signal transfer speed. Okay. So live cells, they have a membrane potential. It doesn't have many functions for that. Like the speed is 5. Like the speed is 2. Okay, this is about the mitochondria. We learned and we know, we think we know, mitochondria has a double layer, double of a double layer. Nucleus also has a double of a double layer. And chloroplast also has the same thing. And if you look at the structure, we draw like this way. If you draw a little bigger, mitochondria structure, one membrane structure here. Another membrane structure is inside here. Each of them is repeated by layer like this. And each of them is a closed membrane. Okay? And if you look at those mitochondria space further, so this is outer membrane. This is outer membrane. This is inner membrane. And here, this is cytosol. So this is inner space. And here, inside here, we call matrix. And here is the intermembrane space we call. Now, let me ask you. Intermembrane space is the inner space or outer space? Here, this is some complicated thing here. So let's look at here and think. Inner space here, is that inner space or outer space in our perspective? It's outer space? Sure? Okay? It's an outer space. Then how about the inside the matrix? Is this inner space or outer space? Is it outer space? Then outer. This is the inner space, no question. This is cytosol. Right? Intermembrane space, topologically, it should be outer space. It is touching is through this membrane, this is outer space. Then, how it is? Matrix. Is that inner space or outer space? It's an inner space, but it's not outer space. It's symbiosis of ancient bacteria space. Right? That's inner space in a sense. That's inner space of life. That's the truth. But that's not our life. That's our partner's life. Interesting, right? They live inside our cell. We say it's a symbiosis living together, but they carry their own space here. And they have their own genome there too. Their genome and our genome is different. Mitochondria does not have a nucleus because it's bacteria, originated from bacteria. And this space, inner membrane space, is kind of DMG, like a fusion space. We don't want to mix up with the bacteria. We have our space, and it's your space. This is the outside space in the middle. That's the situation. Okay? That's why this membrane is outer membrane. Outer membrane is similar to ER Golgi. It's outer membrane. Here, this membrane is bacteria's membrane, not outer membrane. That's why the protein composition here and here is also different. If we draw those structure here, okay, seems like this is one of our organelle, and just a double layer. But if you look at deeper, and actually it's a funny situation. We keep the bacteria in our cells. That's a seal that characterizes the remain. Right? That's the real situation of structure of our cells in terms of mitochondria. Now mitochondria, that's in last week we have studied about photosynthesis a little. That happens in chloroplast, and then using energy by putting the water and CO2 as a source, and then we generate sugars, glucose, and as a byproduct we generate oxygen. This is the process of photosynthesis, and along the line this is the chemical the formula for that. And this is the photosynthesis, and then we generate ATP. In mitochondria we have almost a similar ATP synthesis like this, almost the same structure. So then depending on mitochondria was invented first or chloroplast was invented first, with one started this system. In this case, using photon energies generate this proton gradient and utilize this to make ATP. By the way, this ATP synthesis is the 1/3 circle. Every circle is rotating like this. And then every circle, one molecule of ATP synthesized this side. At least by looking at those shape, okay, we have here, we have here, that is ATP synthesis inside. And this structure is almost the same in mitochondria and chloroplast. That means it was invented by one first, and it will be shared by another. Structure is almost the same. And function is almost the same too. Utilizing this, the proton gradient is generated. Now, chloroplast. Now, if we make it easier, chloroplast was also bacteria. Just imported into our body. This bacteria's expertise is capturing light energy, making glucose. It's a very super excellent ability, but it is very costly too, very expensive. So it doesn't want to do anything else. I just want to make the glucose. And it's imported by outside here. This is plant cells cytosol. And inside here, this is bacteria's inner space. There is intermediate space here too. And that intermediate space is utilized to keep proton gradient, high proton. Mitochondria is same. The membrane space is a high proton. High proton means acidic or basic. Acidic. That intermembrane space is a high proton area. So very similar structure you can see. Okay, so this is the chemical structure I didn't show you last time. This is ADP. And then here, by adding this phosphate, utilizing proton gradient, it can be converted into ADP to ATP. So this is chemical structure for the proton. NAD and NADPH. NADP and NAD almost the same molecule. This is during the photosynthesis utilized. This is for mitochondria that's utilized this. Very similar, only except the difference is with this part. But reaction site is at the top here. This is oxide form. This form is reduced to this form. This is a high energy form. This is low energy form. By capturing those light energy, electron excited, and then that energy has been used to reduce this. This is a high energy form. The 1 ADP H can make a 3 ADP, right? So then this is the closure form. So that means this is the reaction part. This is totally independent of that. That is the help to make localization of the molecules. NADP, NAD, almost the same molecule. Means photosynthesis process and mitochondria process is sharing a lot of common things. Photosynthesis, the energy came from sunlight. Mitochondria, the energy comes from glucose. Photosynthesis make glucose and save them, and then mitochondria utilize it in a reverse reaction, break it, and then generate energy. That is the process. And in both sides, mitochondria, ancient bacteria, chloroplast, ancient bacteria, it's a different bacteria, but they share very common machinery. Okay, now let's think about the nucleus. Inside the nucleus space, is that inner space or outer space? It's a double layer over double. Mitochondria, or the chloroplast, it has closed outer membrane, closed inner membrane. Right? Nucleus, if you look at the student's presentation carefully, she drew like this. Nucleus. One layer, another layer, outer layer, inner layer, nuclear core. This nuclear membrane, if you look at that, is that closed membrane? It's open membrane. It just has a hole. And then the inner space inside the nucleus, is that inner space or outer space? It's the inner space. Should be. Nucleus will keep our gene in there. Most important information. Even the whole other part of the cell dies, if you keep that DNA, it can be utilized later by cloning. Right? It carries the information of life. Right? That should be in a space. And that structure then, how does it form? Mitochondria or chloroplast, it just came from other life, and we just encapsulate it by another member. Right? Then that's why it is a closed membrane structure. Nucleus, well, most likely, it was engulfed in some membrane structure. It could be endosome, it could be this kind of a membrane structure like ER, and they could form this kind of a surrounding structure like this, possibly. And then somehow it has a stable structure. So very funny, nucleus is not closed form. It has membrane structure, outer membrane, inner membrane, but the whole is made, this is connected space to the cytosol. Inside here there are DNA, and RNA is made here, and then RNA is transported. RNA is huge molecules. That's why it can go through this nuclear pore. And the nuclear pore is quite big, maybe 100 micrometers, so maybe half a micrometer. That's quite big, the structure, and it can pass it through these pores. Okay, this is general summary of the cell structure. Last week we said inside and outside of all bodies. And real inside is the inside the cell is the real inside of the cell. Bloodstream is relatively more inside than intestine, but that is still outer space. Gastrointestinal pathway here, that's really outer space. If you just inside out like a donut, it is outside the outer space. That's why we can make it in there. By putting in, just turn it over, right? It's a really outer space. Okay? Real inside is inside of this membrane structure. Very funny, we have some outside material in our cells. Okay? That's the structure of the nucleus, the cell. And I gave you several questions about the concentration, size, and student like. If I can spend a whole other hour to practice with this, but I'm going to remember it. So in this case, how long it can pass through? If it's a bouncy, the light can pass through. What is the speed of light? I hate the kind of 8 or something, so use common the light language. 300 million. 300 million meter per second. 3억 meter per second. or 30만 km per second. This is one of the universal constant that's good to memorize. And how fast it is, sometimes we say it can go through the Earth how many times? 7 times? About 7 times go around the Earth. If it's a city size, then it can be approximately 0.2 milliseconds, assuming that font is so big, font is 63 vessel. I think it's about 10, 20 milliseconds. But anyway, it's that exact number is not important. So amount this amount, this is second. 1 millisecond is a 1 over 1,000 seconds. Even smaller than that, 0.2 milliseconds, light can pass through so quickly. It's much quicker than non-contact temperature. Non-contact temperature usually is about 0.1 seconds. We feel that's very fast, but it's still much faster. If you count that as a cell size, if it passes through, wow, it's a femtosecond range. Now we don't have a good idea what's the femtosecond. That's why I gave you this question. In 1 nanosecond, how far the light can go? This is good estimation about meter. 1 nanosecond is the light can go this much. That is 1 nanosecond. Right? In cell size, it's a femtosecond range. Even in the middle, there is the picosecond there too, right? So nanosecond, 1,000 times lower, picosecond, and femtosecond. Okay? In 1 nanosecond, light can go this much. This is good to know too. Then we can get an idea of what is the nanosecond range. Light can go this much in 1 nanosecond. Femtosecond, you can pass it through only 1 cell size. This is really too small, you can visualize. This is the easy to visualize. This is size of 1 nanosecond. Okay, by using similar, by the way, this is Yevin. Is it yours? It's very good writing, isn't it? I was very impressed. If you can donate your talent to the class by making the class notebook, it would be benefit to everybody. Will you consider? Among the whole homework, this was the best I could see. Almost like a painting. And you can develop like a, Lenin check. You can commercialize too. Think about it, okay? Yeah. So, utilizing this, the analogy, let's think about the size of the cells. Cell size about 10 micrometer. And to make it a better the comparison. Membrane thickness is 4 nanometer. Let's make this into billion times bigger. Billion times 10 to the minus 10 to the 9th. Then 10 micrometer becomes 10 kilometer. 4 nanometer becomes 4 meter. 10 kilometer about the farm city size, let's see. 4 meter is here to here. This is 4 meter. This is the thickness of a membrane. Now if you think this much of a barrier covering whole farm city, the barrier, do you think it's thick or thin? It's quite thin, right? Now let's just think about the penetration of a molecule goes through. Molecule, if you usually, big molecule is like a half nanometer. Half nanometer is like this size. Water molecule like a 0.3 nanometer like this. This is a water molecule, let's say. This water molecule needs to pass through the membrane from here to there. Negative charge here, another negative charge there. In the middle, this is lipid area, right? Now, if it should go through from here, and neutral water molecule go in, touch, no problem, no electric problem. Now, it needed to go this much, may not be easy, right? So to passively penetrate this molecule should not be strongly charged. Charged molecules are super hydrophilic. It cannot survive in the lipid area. It just pumps out. To do this molecule needs to be reasonably hydrophobic. If it is too hydrophobic, like a cholesterol cholesterol cells may be like this. Goes in and here, and it's too heavy inside here. I don't want to go anywhere. Too hydrophobic, it just meet any membrane and just stay there. Or just move this way, not cross that way. That is what happens when we have a very bitter drug. If we take that drug powder, we cannot wash it out even though we drink the water, because it is stuck to the membrane. Too hydrophobic. Similar things for the capsaicin, the spicy one, hot one, the water cannot remove that. What can you use? Milk is relatively more hydrophobic. You can dissolve it and then you can swallow. So to penetrate the membrane, now we are going to talk about later drug. Drug molecule somehow it should go through from environment to the inner water. If not, it doesn't work. Right? So to penetrate this much, the drug molecule needs to be reasonably hydrophobic. Not too hydrophobic, not too hydrophobic. Then water. How can you take the water inside? Water is very hydrophilic. From here across the depth, it's very challenging to the water. We need to have transport of the water. Water channel or water pump. Without that pump, even inside the water, we will die with the crisis. Because we cannot take the water in. Okay, so today's topic was structure of life in terms of cells, cell level. Okay? Then we move to, what is the next week title? I went to Istanbul and just came back early this morning, so I couldn't have a chance to look at next week. What is the title of next week? Molecular life. Okay. Next week, we are going to think about the more molecular level of molecules of life in terms of DNA, RNA, protein, a little more molecular level. Okay, any questions? Okay, we have 1 minute now, so we should be close. And if you have any questions, you can come to me. Okay, by the way, the presentation so far, I asked personally the design for the preparing this kind of thing in the middle or in the beginning. So we have a little bigger number of students. So we have only 14, 16 classes. So it may not be everybody has a chance for the presentation. So by looking at those the topic, you can volunteer and you can send an email. I wanted to make some the small presentation. So you can contact me first. And then first come, first serve. Okay. If I don't have any volunteer, then I will choose